Hello guys, welcome to my YouTube channel. So today we are going to discuss Lac Operon. Now what is a Lac Operon? Lac Operon is a set of genes that are specific for uptake and metabolism of lactose, and it is found in E. coli and other bacteria. Now if you observe the structure of Lac Operon, so first you will notice that Lac Operon contains a set of genes which are closely spaced together. These set of genes are called as structural genes. In case of Lac Operon, these structural genes are of three types encoded by three different genes. One is the LAC-Z gene, alright, which codes for the enzyme beta galactosidase. Then we have the LAC-Y gene. This structural gene codes for the enzyme galactoside permease. And then we have third one, the LAC-A gene. This structural gene codes for the enzyme transacetylase. Now the beta galactosidase enzyme coded by LAC-Z gene is important for the breakdown of lactose. This galactoside permease encoded by the LAC-Y gene is important for the entry of lactose into the bacterial cell. And the third, this transacetylase enzyme encoded by the LAC-A gene. This is an enzyme transferring an acetyl group from acetyl-CoA to galactosides, lactosides or glucosides. Now apart from these three structural genes, some few base pairs upstream, we have an operator for the Lac Operon. Then we have a promoter for the Lac Operon where the RNA polymerase binds to initiate transcription. So this together, promoter, operator and the set of structural genes, this together constitute the Lac Operon. Now some few base pairs even upstream from the Lac Operon, we have a regulatory gene. Alright, now this regulatory gene has two components. One is the LAC-I gene which codes for the regulator, and some upstream of LAC-I gene we have the promoter for this LAC-I gene. So this LAC-I gene has its own promoter. This LAC-I gene codes for a repressor protein in general. So this is the structure of Lac Operon along with this regulatory gene. Now, what happens? Suppose you have a media where you have lactose as the carbon source for bacteria. So the bacteria needs to break down that lactose in order to utilize it as a carbon source and in order to utilize it for its energy. So what happens? Lactose from outside the cell with the help of this integral protein or the enzyme galactoside permease, this lactose from outside enters into the inside of the cell, and as soon as lactose enters inside the cell, Lac Operon becomes active and Lac Operon codes for this enzyme which is beta galactosidase. So this beta galactosidase breaks down lactose into its monomer components galactose and glucose, and in this way the lactose is metabolized within bacterial cells. So now let us see two cases. One is where lactose is absent in the media. So what will happen to the Lac Operon? Now when lactose is absent in the media, this LAC-I gene codes for this repressor protein which becomes active and it binds to the operator region. As a result, what happens? When RNA polymerase sits on the promoter, it cannot proceed further with the transcription because of the blockade of the operator with this receptor. So transcription cannot proceed further in this direction when lactose is not present in the media. Now let us see the second case when lactose is present in the media. What will happen if lactose is present? So as I have already told you, this LAC-I gene codes for the repressor protein. Now if lactose is present, this lactose will bind to the repressor, and the binding of lactose to the repressor makes the repressor inactive. As a result, this repressor is no longer able to bind to the operator. As the repressor is not able to bind to the operator, RNA polymerase which on binding to the promoter can easily proceed downstream and proceed with the transcription. As a result, all the enzymes. All the structural genes are transcribed, and all the enzymes like beta galactosidase, galactoside permease, and transacetylase are coded. Now, this galactoside permease makes the entry of lactose from outside into the inside of the bacterial cell possible, and this beta galactosidase breaks down the lactose into glucose and galactose for easy metabolism by the bacteria.